These develop from what? The other modified develops from microspores, exactly right. Right, now there's an adaptive advantage to having such a reduced gametophyte. You don't have to put as much energy into making it. And honestly, they don't need to, because they don't have to have these gametophytes last as long as gymnosperms pollen grains do. Because in angiosperms, fertilization happens pretty quickly after the pollen gets released. And so the pollen doesn't have to last for very long. That's not the case in gymnosperms. There's a little delay time there between when pollen actually gets released and when fertilization actually occurs, so that pollen grain has to last for longer. They have to put a little bit more energy into it to make it a little bit more developed. Now we're going to get into our female gametophyte, and we start talking about how that one is also going to be reduced. It's going to be made up of 8 nuclei, but only 7 cells. Okay? And so here you can kind of see where these 7 cells are located right here. We've got 3 on the top, we've got 3 on the bottom, we've got one big one. That's all the rest of that volume right there. And the reason there's only 7 is because one of those, the big one right there, actually has 2 nuclei as opposed to a single nuclei, which all the rest of them have. So if you add those up, we've got our 3 nuclei here, 1, 2, 3, 4, 5, 6, 7, 8. Those are our 8 nuclei, but 7 cells total. And these gametophytes are so reduced that the angiosperms have actually lost the archegonia feature because it can't form in such a small structure like this. But the ancestor did have that. So here's how it forms. This is what we were just starting to get into. In some ways, there's going to be some similarities here between what we saw with the conifers and their life cycle, where I talked about, okay, here's what the female side is doing, here's what the male side is doing. Microspores are developing in pollen grains. Still works that way for this one. Okay? And our megaspores, they are developed into female gametophytes, but not all of them. Right? And the same thing applies here for our angiosperm gametophytes. In this case, a cell in the megasporangia divides by mitosis, okay, to form 4 megaspores. Okay? And the 4 megaspores, 3 of them are going to disintegrate, okay? And only 1 of them is going to develop into the female gametophyte. Now, that one functional one, you can see it right here. So here's our 4 megaspores. These 3 are going to go away. They're going to degenerate. This one right here is the only one that's going to develop. It will become the female gametophyte. So it's going to kind of fill this space up here and become what you see here. Okay? So the one functional megaspore undergoes 3 mitotic divisions to form a female gametophyte with 8 nuclei. If you think about that, that makes sense, because if mitosis occurs, you're dividing each time by once, right? So we've got a single nuclei here, we divide it once, we get 2, right? You divide it again, you get 4, right? And you divide those, you're going to get 8, right? So we end up with 8 total in terms of nuclei. And then those nuclei arrange themselves into the 7 cells. Now, what those cells are, there's actually only 2 that are really important that I want you to know about. I'll show you where the other ones are, but we're only going to discuss the importance of 2 of them. So you've got your egg cell. That's the first one I want you to know. That one's located right here. So that one's obviously pretty important because that's what's eventually going to become the new baby plant. It's flanked by 2 other cells right here. They're called synergid cells. You don't need to worry about them. Essentially, they are going to be, one of these is going to get ruptured when the pollen tube comes down here and releases the sperm, so they're kind of unimportant. Okay, so those 2 flank the egg cell, but the egg cell is sitting right there. Then you have 3 cells on the opposite side up here called antipodal cells, which their function is not completely understood, so we're really not going to talk about them either. But we are going to talk about the big one. And then you have the remaining volume here, which is referred to as the central cell. Okay? And it is the one that has the 2 polar nuclei. So those are the 2 I want you to know: the egg cell and the central cell. Right? And I know there's a lot of cell names here. Okay? Don't get the generative cell and the tube cell and the male gametophyte mixed up with the central cell and the egg cell and the female gametophyte. They have different terms for them and they do very different things. But both of these are actually going to be involved in the fertilization process, and both of these cells are actually going to get fertilized by sperm. Hence this term that we're going to get into here shortly, which is also a shared derived trait for the angiosperms, which is double fertilization. Okay, so we've got double fertilization. It's a bit different than the double fertilization that we saw in the details. This is going to be considered still to date a shared derived trait for this group because it's unique and thought to evolve separately from the double fertilization that we see in the details. So what happens after pollination occurs in an angiosperm is you get your pollen grain up here, okay, it sticks on the top of the stigma, the pollen tube is sent down to the bottom, all the way down here. It's going to burst one of these two synergid cells on either side of the egg cell here, and it's going to release both of the sperm into this area right here. So remember that generative cell is making two sperm. Okay, so both of those sperm are getting delivered. One of those, okay, is going to be fertilizing the egg, and one of them is going to fertilize the central cell. So two sperm, therefore double fertilization. And what it makes is going to be super important for angiosperms as well. Okay. All right, so the first sperm is going to do what you expect it to do. It's going to fuse with that egg, and we know what forms when that happens because it's part of the alternation generations life cycle. You get your zygote, and that zygote ultimately is going to develop into what? The embryo inside the seed, exactly right, which will eventually become the sporophyte, exactly right, which is the dominant stage for these groups of plants. So we get our diploid zygote, and it's diploid because both of the gametes, the sperm and the egg, are haploid. So this one's going to be diploid. And I've highlighted that because it's different for this next one, because as you remember, this central cell here has two nuclei, not one. Okay, so the second sperm is going to fuse with those two polar nuclei in the central cell, and it produces something called endosperm. And it will be triploid, because you're taking the set of chromosomes from the sperm, one set, and then you're getting two sets of chromosomes from that central cell, making a substance, a nutritive tissue that's going to be triploid. This substance, the endosperm, is a shared derived trait for angiosperms. It's kind of an important one because it makes up a significant component of the seed. Now there's always questions on the test that relate to the cell types, right, for both the female and male gametophytes, and there's always something in there, fill in the blank, related to double fertilization. So you need to know what each sperm is fusing with and what each sperm and egg or sperm and central cell is making. Okay, so we've got our zygote versus our endosperm. So let's delve a little deeper into the endosperm. Okay. This is the nutritive tissue that's inside the seed. If you recall, seeds have three parts to them. They have the embryo, the baby plant. They have nutritive tissue to supply that embryo with the resources it needs to grow and develop before it's able to get its own water and nutrients and photosynthesize, because its leaves are not developed yet. And then you have your seed coat. Now, in the gymnosperm seed, the nutritive tissue was the female gametophyte. For angiosperm seeds, it's going to be this endosperm. It usually envelops the embryo, and how much of it there is in there and how nutritious the seed is will depend on species. Right? It forms through the process of double fertilization, which means you don't get it unless fertilization occurs. That's actually going to be one of the adaptive advantages of the endosperm that we're going to talk about. So this is going to replace the female gametophyte as the primary nutritive tissue for the embryo and the seeds, rich in carbohydrates, oils, proteins, all things that that developing embryo is going to need to survive. So now we're going to talk about adaptive advantages here for our endosperm. And there are going to be two main ones I'm going to discuss. Advantage number one. Remember, this does not form unless fertilization occurs. So you have guaranteed here that an offspring has been produced, and the embryo is a baby plant. It's something that can potentially develop into an offspring. Whereas an unfertilized ovule is not necessarily going to form into an offspring unless it does get fertilized. So we don't make the food for this embryo until it's needed. Right? You know you need it now because this egg has been fertilized. It is going to develop into an embryo, so it is going to need this nutritive tissue. So we're not wasting any resources or energy forming nutritive tissue for ovules that may never be fertilized, which is what the gymnosperms do. And there's no guarantee that that seed is actually going to get fertilized. And if you think about what we discussed in terms of the gymnosperms, the female gametophyte, because that's the nutritive tissue, that has to form before fertilization happens. Think about alternation of generations. You got your sporophyte, makes the spores, forms into the gametophyte. That forms first, and then the gametes are formed and fertilization occurs. So gymnosperms, since that's their nutritive tissue, they're gonna have to make that regardless. They don't even know if it's gonna get fertilized yet, so it's kind of a waste of energy for the ovules that don't get fertilized. But we don't have to worry about that for angiosperms, because it only forms if it gets fertilized. Second, remember that it's triploid. It has 3 sets of chromosomes instead of 2. And remember, just because you have the same chromosome that have the same genes on it, doesn't necessarily mean you have the same alleles for that genes on it. So you get a little bit more variation here you can have in terms of what these nutritive tissues could be, and what these carbohydrates, what these oils, what the things that are that it can make in terms of a food source. And it also may be able to develop more rapidly too, because it's triploid. I mean, an extra set of instructions can help with that. But this is the bigger one, the greater potential for chemical variation and nutrient contents in terms of what they can supply as food. More options there. All right. Now, the last shared derived trait we're going to talk about for angiosperms is something that we have discussed earlier when we were talking about vascular tissue, because we talked about both xylem and we talked about phloem, and we sort of hinted at the cells that were involved in those 2 types of tissues. We really discussed xylem in detail with tracheates and basal elements, but I told you we'd come back to this again for the phloem and discuss the specific cells in a little more detail when we got to this chapter, because there were 2 types of cells that are related to the phloem. You have seed cells and you have seed tube members. That was your bonus question on your test. Not everybody got that one, right? But that's what those 2 cells are. It's listed back in that previous chapter related to vascular tissue. Now, seed tube members are the more involved of the 2, and they are only found in angiosperms. Hence, this is a shared derived trait. So we know that since these make up phloem, and phloem is the vascular tissue that moves the sugar, these are sugar-conducting cells. And they're responsible for moving the products of photosynthesis from the places where photosynthesis is occurring, like leaves, and the green parts up here, down to the places that can't photosynthesize, like the root systems. So they're going to make up what type of vascular tissue? Phloem. Exactly right. Okay. Only found in angiosperms, hence we can call them a shared derived trait. And they are evolutionarily modified from the more primitive cells, which are the seed cells. So just like vessel elements evolved from tracheids, seed tube members evolved from the seed cells. But this is going to be a little bit different than what we saw for those xylem cells. Because in the case of xylem, and the tracheids and vessel elements, either you have tracheids only, that was the option, or you had both. You had both tracheids and vessel elements. Those were the 2 options, either one or both. Right? And you can't have a situation where you have just vessel elements. That doesn't happen. It's very different for these phloem cells. Right? You're either going to have seed cells or you're going to have seed tube members. You don't have both, ever. You either have one or the other. And angiosperms have the seed tube members. That means that anything else that is a vascular plant is gonna have the other, the sieve cells. So let's compare the two. In sieve tube members, you've got pores on the end walls that are larger than the pores on the side walls. One of the characteristics we talked about for the sieve elements, all the different types of phloem cells, is they're elongated, long and skinny. They form into pipes and tubes, just like the xylem cells do. And they have these collections of pores here, which is where the sugars get pumped into the cells. Right? This is kind of the lateral transport here, getting it into the cell, and then you have these pores on the top and the bottom here that are connecting into the next cell above or below it that are responsible for moving it vertically, okay, downward. Okay? Now, in the case of these sieve tube members, you can see that these are small little pores, but the ones that are on the tops and the bottoms of the cells are bigger, right, than what they have on their side walls. So that's how you can tell these are sieve tube members and these are sieve cells, because the collections all look the same here. These are bigger than the ones on the side walls. Second, okay, there's a name for these collection of differentiated pores on the top and the bottom of the sieve tube members. We call them sieve plates. So these are not called sieve plates. These are called sieve plates. And here you can see the plants that have them. Make sure you know them too, right? You should be able to fill in a table. We had a table on the test that talked about different types of cell types. We talked about what their functions were. We talked about whether they were alive, dead, semi-alive, whether they had primary cell walls only, both primary, secondary cell walls, what kind of tissue they were a part of, and we could also talk about what groups of plants they're in, right? So where do we find Basal elements? They're in angiosperms, or some of them, and they're in the tails. Okay? So where do we find the tracheates? In all vascular plants, right? So in the case of these, you only find the sieve cells in sieveless vascular plants and gymnosperms. Pretty much all the vascular plants that are not angiosperms, that's where you find them, okay? And you find the sieve tube members only in the angiosperms, okay? Now, one other big difference about them is these extra cells that are going to be right next to the sieve tube members that function in loading and unloading the sugars, that are called companion cells. Now, sieve cells are going to have cells next to them that do this job, but they're not actually derived from the same cell that the sieve tube member is. These are, okay? So this sieve tube member and the companion cell that would be right next to it came from the same cell. They were both made from that same cell. And so we don't call the ones related to sieve cells companion cells. We call them albuminous cells. They're different. Okay, so these are parenchyma cells. As you know, those are the most versatile of all the plant cells. They can do a lot of different things. They're responsible for photosynthesis, cellular respiration, wound healing, regeneration, but we also heard about how they're responsible for storage of nutrients and lateral transport, right, of substances, and this is where that comes into play here, right? So these companion cells that are parenchyma cells, right, are supplementing for some of the functionality that the sieve tube member has lost because it's only semi-alive. Sieve tube members, sieve cells, semi-alive, they retain a functioning plasma membrane, but most everything else inside of it is non-functional. And they don't have the nucleus, ribosomes, things like that. What little few organelles they have left are all kind of shoved off to the side, and so they don't really have any basic cell functions other than to move these sugars. But the companion cells are complete cells, completely functional living cells. And so they have those ribosomes, nuclei, they can serve some of the functions that the sieve tube member can't. But the biggest function they have is to load and unload those sugars into the cavity. Because unlike water in the xylem, which is going to flow the direction it wants to flow without using any energy to do that, this is going to require a bit of energy and effort on the plant. This is not where the sugars want to go, because solutes don't tend to want to be concentrated. And that's exactly what's going to happen in the sieve tube members. You're going to shove a whole bunch of sugars in here. Now you've got a really concentrated solution, which is why the water comes in, but it doesn't want to stay like that. And so there's some active transport that's involved in here for putting those sugars in. And again, the companion cells form from the same parent cell as the sieve tube member, so these both came from the same cell, whereas the little cells that are next to the sieve cells don't. Okay, distinctive line here, okay? Everything we've discussed so far are shared derived traits for the angiosperms. They define that clade. They are found only in angiosperms and no other groups. This next one is different, right? But I want to still talk about it because all angiosperms do have these features, right? And this particular one we're going to talk about is going to be Basal elements. Whereas the seed tube members are in fact shared derived traits for angiosperms, we can't say the same thing for Basal elements. Not all of them are going to have Basal elements. Some of the more primitive families, like the water lilies, don't have these. And we'll find out, we also found out that there's a group of gymnosperms that actually has these as well. But these are water conducting cells, so these are going to make up xylem. And if you have Basal elements, you will still also have tracheids. So what was that other group that had them? Which one? No, vascular plants, no.
Knee-tails.
Knee-tails, exactly right. Specifically the knee-tails. Okay, that group, monophyletic group within the conifers is going to be the other group that had that. And they were unique too compared to the angiosperm ones because they have the porous holes on the top and the bottom of the cells. When you get to the ones that are in angiosperms, those holes tend to be more like bars or slits or just one giant hole altogether. So it is possible that the Basal elements that are in knee-tails and the ones that are angiosperms actually were independently derived. They have not figured that out yet. Right? And that's why the knee-tails are so hard to place because they're in some ways very much a sister group to the angiosperms. They have double fertilization, they have Basal elements, and yet people are still thinking that they're supposed to be the conifers and are evolved all these characteristics separately. Now, the adaptive advantage of having Basal elements, if you have them, in addition to your tracheids, is that they are bigger tubes, right? They've got bigger holes that actually go all the way through the secondary cell wall in addition to the primary cell wall. And so they're going to move water much more efficiently. Just like a little coffee straw versus a big giant Slurpee straw. They're going to be more efficient in moving that. That's one of the reasons also why angiosperms are the dominant species today, or the dominant group today, because they're pretty much better at everything. They have all these extra adaptive advantages that other plants, even the gymnosperms, don't have. Okay, now that's going to be our intro to angiosperms. What we're going to be doing next for the rest of the semester is we're going to be going into the different families of angiosperms that we're going to talk about. We're definitely not going to be able to hit all families in the angiosperms because there's just way too many of those. So what I'm doing is I'm picking the most common ones, the ones you're likely to see around here, and the ones that are definitely economically important as well. So we'll get into some ecobotany as well. We're going to start with what are called the basal angiosperms. Okay, so here's our cladogram for the angiosperms. We're just taking that one clade, we've removed all of the gymnosperms, all the other vascular plants and the non-vascular plants from this. We're just looking at the flowering plants. And you can see most all angiosperms fall within this grouping right here, which is going to include something called the monocots, and all of these, which are called the eudicots. So we're going to spend a lot of time talking about those. But you'll see there are some more primitive lineages that are down here. And that's what we refer to as basal lineages. When we say basal, we mean primitive, right? The primitive groups. And so many of those are not necessarily going to have all the adaptations these other ones do, including things like Basal elements. Okay? And this phylogenetic tree is still a work in progress. They're still trying to figure these out. It was changed since the second edition of the book that you have as well. We're not going to discuss all of these. Right? So for example, we're not going to do pepper rails, which is where the pepper plants are in. But we're going to focus on the three that are circled here. So the nymphales are Magnoliaceae family, which is the magnoliales and the laurels. Monocots actually used to be considered basal angiosperms. They are not anymore, though. Alright, so our first family we're going to discuss is Nymphaceae. I will always give you the scientific name and the common name. Make sure you know both, and you can match those together. This is the water lily family. And it's probably the family or the common name that students mix up the most. Because later on we are going to learn the Liliaceae family, which is the true lily family. But water lilies are different than lilies. Right? Very different groups. They're less evolved, more primitive. And this is also one of those families that made that transition to land and then went back again to the water. So they have all the characteristics we expect land plants to have, but they are now going to have adaptations for getting them back in the water, dealing with those sorts of conditions again. Not a big group, 6 genera, 60 species. I'm never going to ask you that. You don't have to memorize those numbers, okay? But I want you to kind of have in perspective the size of some of these families, because later we're going to be talking about like the orchid family that has 20,000 species in it. So a big difference in size. This is a pretty small one. They are all aquatic, hence the name water lilies. And oftentimes these are going to have milky latex associated with them. So if you remember when we were out looking at our weeds, we broke the dandelion stem off. We looked at the peduncle when we broke that off and started to bleed this kind of milky substance that was the latex. So there are certain families that tend to do that, and this is one of those, and it's a defensive mechanism against herbivores. And as I go through the families, I'm going to give you characteristics that relate to the flowers, or excuse me, I'm sorry, the habit, the leaves, the flowers themselves, and in some cases even the parts of the flowers, and the fruits. And you may not necessarily need all those characteristics to identify a plant, because there are certain distinctive characteristics that are more useful than others. So I've tried to kind of highlight and underline the ones that I think are the most important. So if I've bolded and highlighted, you probably want to pay special attention to that particular characteristic. If it's not bolded and highlighted, probably less important. Okay, but I'm still going to give that information to you as we go through it. All right, now people normally think these are floating plants because they see the leaves kind of floating on the surface like this, but that's actually not the case. There are such things as floating aquatic plants. Okay? So frog bit, which is something around here, it's a little small plant like this, is a true floating plant. The root systems are not attached to anything, it just sits on the top of the water and kind of floats its little way with little gas chambers underneath the leaves. But these are not floating plants, they are in fact rooted plants. If you go all the way down to the bottom of the plant, they're going to have to be in shallow enough water that they actually can have their root systems in the soil, the sediment at the bottom of the lake here, and still be able to reach the sunlight on the surface. So they're going to have incredibly long petioles on their leaves to get up to the surface there. The stem will either be tuberous, like you can see down here. Like tubers are essentially little storage product or storage chambers in root systems, or they're rhizomatous. And many of the ones around Reelfoot Lake are rhizomatous. They've got really, really thick rhizomes. They're about that big around, they're very dark brown. And when they actually go in and they kind of, in some of the areas where they try and clear the trails for the canoes, they'll kind of pull some of those up, dig those up. You'll see them kind of floating on the surface, you can collect those if you wanted to. I have a couple specimens of those, but I didn't bring those in today because students always think that they look like fecal material. This is about the size and the shape that they are. Okay? But they do have rhizomes underneath there. They also have simple floating leaves. So even though the plant itself is not floating, the leaves are. They get to the surface on those long petioles, and then they float so that they can reach as much sunlight as possible. Because the deeper you get in the water, the less light you get. Water is going to absorb and reflect that light. And so if you get to Reelfoot Lake, you're going to find these all along the edges of the lake, but if you get too far into the center, they're not there because it's too deep. And they wouldn't be able to be rooted in the soil like they have to be, and still be able to get their leaves high enough that they can actually get to the sunlight. So they're going to be on the shoreline for that reason. They are also oftentimes peltate. Now if you remember what peltate means, that means that instead of your petiole coming up on your leaf here and attaching at the base like we expect it to that way, it comes up and attaches kind of in the center of the leaf, making it look a little bit like an umbrella. So here's our leaf, the attachment point would be underneath like this. It would look sort of like that. So it's common for these to be peltate. Okay, flower-wise, They have solitary flowers, so they're not gonna have panicles, racemes, spikes, or anything like that, just a single solitary flower on the stalk. And it is emergent or floating right on the surface. Reason for that is because these do not use water pollination. Water pollination is a primitive method of pollination. It is risky, it is very chancy as to whether or not your pollen will get to where it needs to be if you're just gonna rely on the currents of the lake or the ocean for that matter, if we're talking about another type of aquatic plant, to get your pollen to your flower. So many aquatic plants nowadays actually are insect pollinated. These are in fact insect pollinated, which is why they bring those to the surface. And ecology, plant ecology, my students learn about one that has giant flowers that open up just twice. And it opens up one day, it attracts beetles to it, it closes up on them and traps them for 24 hours, and then the next day it lets them back out again after they disperse that pollen thoroughly throughout the flower, lets them go, and then it goes back down into the water. They are perfect flowers, meaning they're bisexual. They have male parts and they have female parts on the same flower, and they're both functional. And the flowers are actinomorphic. Now, one thing I will caution you about for the flowers here is not everything that has nice little round lily pad looking leaves like this out at Real Lake are things that are in this family. There's another species out there called water lotus, which is in Numbaceae, which is a different family. Okay, so not everything is a water lily that looks like a water lily. These are the characteristics you have to actually have to be in this particular family. Okay? You have anywhere from 4 to 6 sepals and lots of petals. But if you go into the center of the flower, as you get further from the outside and move towards the center, your petals start to look weirder and weirder and weirder until all of a sudden they look like stamen. Okay? So what they're doing is they're grading into what are called laminar stamen. So this is what the outermost ones look like. As you get closer towards the center, you're actually going to see theca, or pollen sacs, start to form on the tips of the petals right here. And then as they get further inward, this bottom portion of the petal starts to look more and more like the filament until it's essentially the filament. Right? And we'll have a couple families that do this, that have these laminar stamen. So I'll talk about that in a little more detail here with the Androecium. Laminar just means that those stamen are going to be flattened, right? Not three-dimensional way you expect pollen or a stamen to be. They're flattened and more petal-like or leaf-like. So essentially it looks like you got a petal that has pollen sacs on it, is what that looks like. Now for some species, their laminar stamen all look the same. Right? They're all the same flatness, the same size and shape. But for the case of Numbaceae, if they have this, it does become more pronounced, right? And more stamen-like as you get towards the center. So it has kind of a gradation to it. Magnoliaceae, which we'll learn later, doesn't have that gradation. It just has the flattened stamen, and that's it. Okay. Now the fruit. It's actually a capsule, but it's a weird looking capsule. Because capsules are known to have multiple carpels and they're known to be dehiscent. Meaning, if you talk about dehiscence, usually it means you've got some sort of regular pattern here. That this particular fruit type pops open along sutures or seams, right, that are typically always in the same place. These are a little bit different because they dehisce kind of irregularly. There's not necessarily a set suture this is where it's going to pop open. It kind of pops open wherever it wants to, wherever it's weakest on that capsule. So it's kind of fleshy and spongy and pops open irregularly. This one we're going to talk about. And you can kind of see how this side decided it was going to come open on that one. Right, this one kind of popped open more on this side right here. But we'll look at this one in more detail here in a second. Because I always like to give you at least some examples of some of the plants around the area here that fall within these families, in case any of you run across them and decide you want to put them in your collections. So here's one called Spatterdock, Nuphar advena. You'll definitely find this around Reelfoot Lake. It's probably the most common one that's out there. Their leaves are a little bit less rounded, so we're gonna look at another species in just a minute that has leaves that are a little more kind of heart-shaped, like this. A nice little wedge there. These are more arrow-shaped, so they're much more long this way, and then they'll have their wedge here. And they have these little tiny yellow flowers like this that are about the size of a ping pong ball. That's the best way I find it, just with the little yellow ping pong balls that are going to be emergent. They stick up a little bit above the water when they're actually in flower. And then they produce those little fleshy, spongy capsules that you see right here. But probably the most common one. But there's also another one out there that is Nymphea odorata, which is the white water lily, which is less common. It's the one that has the leaf that's more like this one. It's named Nymphea odorata because it smells wonderful. It has a wonderful odor associated with it, nice and sweet. And you'll find them kind of in clumps in certain places, so in higher abundances in certain kind of occasional spots, but not spread throughout the entire lake like the spatterdog is. And it has white flowers that are much bigger, and the lavender steam are much more obvious than the spatterdog's little yellow flowers that are like ping pong balls. And again, the water lotus in this family. So we're not going to talk about that one. So the one that makes that weird looking fruit, you probably guys know what I'm talking about. The fruit that is kind of funnel shaped. This sort of rounded on the top. And then you have these little holes and have these what look like little nuts down in there. That's the water lotus. That's not in this family. All right, the next group we're going to talk about is Lauraceae. This thankfully has a common name that's very similar to the scientific name. It's called the laurel family. You're probably more familiar with plants in this than you think you are. So I'm actually going to talk about some economically important plants and some native Tennessee plants that go in this family as well. And I'll tell you that if I do mention economically important plants, those are ones that you do need to know for the test. You don't have to know the native species like the 2 water lilies I just taught you. Don't have to know those and memorize those. That's just purely for your collection. But if I give you an economically important one, and I'll tell you when it's economically important, those I want you to know. Common names are fine. Okay? Most of these are going to be something woody, either shrubs or trees. So I've got that one bolded for a reason, because we're going to get into some of the families where pretty much all the plants are herbs. And so if it's a tree or a shrub, then you know it's not going to be a lily, for example, or an orchid. The other thing about them is many of them have aromatic oil glands in their leaves, which means that when you crush the leaf, you usually can smell it, okay? And in a good way. Usually they're good scents, spicy or lemony, okay, kind of peppery. And which is why some plants that are in this group are going to be used for culinary purposes, because of those oil glands. Sometimes they can impart some flavor to our food. So we actually have some types of plants like this right in front of the library. I believe they've got the sweet big plants that are planted that you can crush the leaves and smell those aromatic oils. Now most of these are evergreens. Ironically enough, some of the ones we're going to learn, like the sassafras, aren't. They have simple leaves though, that are mostly alternate. And they're either undivided or lobed. So the Persea up here you can see is going to be an entire margin, but sassafras, which we're going to talk about later, is weird because it has trimorphic leaves, which means the leaves can take on 3 different forms. They can look nice and entire, like a regular old leaf here. Right? They can look kind of like a mitten, where they have a little thumb sticking out on one side right here, and then the rest of it's entire here, or a lobe sticking off, or you can have something more like this. It looks like something from a Star Wars movie, or Star Trek, where you got the 3 lobes right here. Okay? And we'll get into it. Has anybody actually smelled sassafras before? What does it smell like? Fruit Loops, exactly right. Or if you like Fruity Pebbles. Somebody got really upset because I said Fruit Loops and said Fruity Pebbles one year. Whichever one you like better. It smells fruity, but why? Because they have aromatic oil glands in the leaves. It's just characteristic of that particular family. I like Fruit Loops personally, but somebody like Fruity Pebbles. Now, flower-wise, they're small. They are either perfect or imperfect. So, for example, in sassafras, it has separate male and female flowers. Here's your male flowers, the stamens flowers. Here's your carpellate flowers, the female flowers. But in Persea americana, which we're going to get to that one, this is economically important in a second, That one's bisexual, has both male and female parts on the same flower. They are actinomorphic. And I've bolded this one because I want you to know this one. Usually there are 6 tepals. You can't differentiate between the sepals and the petals. They essentially look the same. They all look like petals. And they'll either be greenish, yellowish, or white, and you may want to kind of circle yellowish. Because around here, most of the things that you're going to find that are in this family, you're going to have yellow flowers. And it's not uncommon for them to put their flowers out before the leaves come out. So sassafras, it's not wrong for you to have in your collection a twig with yellow flowers. I mean, that's perfectly fine, because that's what you're going to find for the sassafras if you find it. Same thing applies to the spice bush. All right, other characteristic I want you to know for sure is related to the male parts, the stamen. Remember when we talked about the stamen being a shared drive trait for angiosperms, of how all of them dehisce in different ways. Sometimes they have little pores and openings on the top, some of them have slits and sutures, and some of them have flaps like this. So if you look closely at these, you can see there's these little kind of cavities in here where the pollen's being housed, but these flaps kind of tear upwards and peel away from it, opening up the chamber on the inside, allowing the pollen to get out. Sometimes it'll have 2, like here, we've got 2 right here. Sometimes it'll have 4, 1, 2, 3, 4, but all of them have these what we call valvular anthers. And I've had students come to me before with plants and say, I've been keying this forever and I can't figure out what it is. And I look at this twig with these little yellow flowers, and I look under a microscope and I see that, I immediately know they got sassafras. Okay, so it's very distinctive. If you can find the microscope and you can look at those and see those valvular anthers, then you know you've got something in this family, and it's going to help you identify it. The gynecium, a little bit less distinctive for the family. You have 1 carpel with a superior ovary that's rarely inferior. But you notice I don't have anything bolded there. Okay, and then our fruit is usually some sort of a berry or a drupe, oftentimes dispersed by birds. So a drupe is going to be one of those kind of fleshy type fruits that has a pit in it. Okay? Although some of them are not fleshy, for example, almonds aren't. But if it's got a pit in it, it's going to be some sort of a drupe. Like our avocado, which ironically enough is actually in this family. Now, fruits are always a good thing to know. Usually on tests, both the last exam and also the final exam have a section that is just nothing but fruit matching. So make sure you know what the common names are for those families, make sure you know what their fruit types are for sure, because sometimes those are distinctive and good diagnostic characteristics for recognizing the family. Here are some of the species that you'll find in Tennessee. Sassafras, definitely one of them. We're going to learn that one later in the semester, so I'll talk about that more then. But we also have something called Lindera benzoin, which is it's a shrub, right? But it is called spice bush. So when you crush these, it has kind of a lemony pledge scent to them, but it's a little bit pepperier and spicier than just lemon pledge. And so it's going to have its flowers come out before its leaves as well. So you may have a twig with flowers on it, just too, if you happen to find that in this. I know I've seen it at Beach Ridge before, so it's definitely out there. And it's got those bright fruits, those bright red fruits that you see there. Economically important ones, well, bay leaves come from these. Okay, so this is the bay laurel right here. So if you've ever used bay leaves for your soups or any of your stews or things like that, it comes from a plant that's in this family, and those aromatic oils are actually contributing to the flavor. Avocados are also in this family as well. So these are economically important ones, you know these for sure. And we will have one more that's economically important, but I'm going to wait until the next class to do that one because I want to spend a little extra time studying it, talking about it. So I'm going to pass the test back. I will also bring in the next class another waiver for you guys to sign.